Welcome to the Pro Tips video series designed to provide biology students with some background knowledge needed to make upcoming lessons more manageable. Students are encouraged to watch these tutorials before attending a lecture on the topic and rewatch them as needed while studying. I'm Professor Workman, and today's Pro Tips introduce enzyme function. The first key term related to enzyme function is enzyme. An enzyme is a cellular machine that regulates a specific chemical reaction. There are multiple enzymes in your body, each responsible for regulating its own unique chemical reaction. Enzymes are made up of proteins, and to rely on this indelicate analogy, you can think of an enzyme as eating its own substrate and performing a chemical reaction to convert that substrate into a product that it then poops out. Our second key term is the verb to denature. Denaturing happens to proteins when they lose their shape and therefore they stop functioning. The process of denaturing is irreversible, and you've witnessed how heat irreversibly denatures proteins if you've ever cooked egg whites. When the runny proteins become denatured, they turn solid and white. I've already mentioned heat's capacity to denature proteins, but are there additional ways to denature a protein? Yes, you cook them. We cook our food for a variety of reasons, but one is that it's made safer to eat by denaturing the proteins of pathogens like E. coli or Salmonella that are found on the surfaces of the food. Additionally, denaturing the proteins in the food itself makes the food softer and easier to chew, like when you marinate meat in citrus juice. So these three pictures show traditional ways of preparing food that is denaturing the proteins in the food. You can apply extreme temperature, extreme heat. The middle panel shows ceviche. In this picture, I see fish and vegetables that have cooked in lime juice, which is acidic. That's an extreme pH. And in the far right panel, I'm showing salt-packed fish. When you subject proteins to an extreme salt concentration, extreme salinity, that denatures the protein as well, potentially. Given that different environmental conditions could potentially denature my enzymes, how do I know if an enzyme is still functioning? Let's return to our indelicate analogy. Imagine the lab scientist is a farmer feeding the cow enzyme its substrate, grass. If the cow enzyme is functioning, that grass will be converted into cow pies, the products of this chemical reaction. In other words, if you have an enzyme and you feed it its preferred substrate, if the enzyme is functioning, it will bind to the substrate, a chemical reaction occurs, and you're left with more products and less substrate each time the enzyme performs this task. If products are made, then the reaction took place, and if products are made at a rate faster than occurs naturally, then the enzyme is functioning. This is an important point. Products may be made very slowly in the body, or they may be made under conditions that are rare in nature, like if they're struck by lightning. Enzymes, though, are speeding the rate of reaction. So if the products are appearing in your experimental test tube faster than they would without the enzyme, then you know the enzyme is functioning. Let me show you what this looks like during an experiment. Consider this graph of experimental results. In this experiment, we have an enzyme assisting a reaction in a hypothetical test tube. And we're using something to detect the amount of product found in the test tube over time. So you'll notice on the horizontal axis, this is time across which the experiment is being monitored from time 0, when the experiment began, all the way up to 15 minutes. And the vertical axis shows the amount of product in the test tube. The units are milligrams. So you'll notice at time 0 there isn't any product because the enzyme hasn't had a chance to work yet. But as time progresses, more and more product is made until the graph flattens off. This is called an asymptote. So what is happening here at this flat part of the graph? The answer is that in the test tube, the substrate ran out. So it's not necessarily that the enzyme stopped functioning, although maybe that happened. We'd have to rule that out in our experiment. It could just be, though, assuming that no extreme heat, no extreme pH, and no extreme salinity were applied, that the enzyme just ran out of substrate. Here is a different graph from that same experiment. The orange line is the same one you've seen before, where the amount of product is increasing over the duration of the experiment up to an asymptote where it flattens off. The new line in blue is decreasing, and this represents the amount of substrate detected in the test tube. And you'll notice that as the substrate declines, product increases, and this makes sense because the task of the enzyme is to convert substrate into product. In an experiment, or perhaps in a medical scenario, you might want to inhibit a specific enzyme's function. So what are some ways that we can inhibit the functionality of an enzyme? You guessed it, we can apply extreme temperature, extreme pH, or extreme salinity in an attempt to denature or at least slow down inhibiting the function of the enzyme. Consider these graphs of experiments on the same enzyme at different temperature levels. The first graph on the left shows a normal functioning enzyme like we've seen before. This is at body temperature, 98.6 degrees Fahrenheit, which is about 37 degrees Celsius. Contrast the function of that enzyme at that temperature level with its function at a different temperature level. So on the right, we have the same experiment, except we've heated the test tube to boiling. So 212 degrees Fahrenheit is the same as 100 degrees C, and this is the temperature at which water boils at sea level. And here, notice the amount of product detected in our test tube remains 0 at all time points. What happened here at this high heat? Hopefully you guessed that the enzyme denatured. It was cooked in the heat. On this slide, we're showing something very similar, but the example is extreme pH. So here we have on the left an enzyme functioning normally at pH 7, but on the right, that same enzyme is not functioning at all at a more acidic pH of 5. What's going on here? Well, the enzyme denatured due to the extreme acid environment. Finally, the same graphical comparison would be made at different salinity levels. So on the left, we have a 0.9% salinity concentration, which is roughly identical to the saline concentration of human blood. And then on the right, we have a 5% salinity concentration, which is very, very salty. And that extreme salinity denatured the enzyme, and that's, again, why we have no product made across the 15 minutes of the experiment in the 5% salinity scenario. Here's a thought question for you to consider. Do you think all enzymes in the entire human body have the same preferences in terms of temperature, pH, and salinity? Well, here's a hint. Some enzymes live in the stomach, which can go down to a pH of 2, which is very, very acidic. But other enzymes can live somewhere else, like in the intestines, that routinely has a pH of 8, which isn't acidic at all. In fact, it's basic. Do you expect these enzymes to have the same preferred pH? Or how about this second example? Think about the enzymes operating in freshwater fish versus saltwater fish like Nemo here. Do you think they have the same preference for salinity? So the answer to the thought question is no. Each enzyme functions optimally under its own set of conditions, often matching the temperature, pH, and salinity inside the cell that produced that enzyme. So let's say you have an experimental enzyme that you don't know anything about yet. How do I find out what environmental conditions suit this enzyme? And you may actually do this in your biology class. You're going to run an experiment. So here I'm showing multiple test tubes, and let's run this experiment in our minds. Our experiment is going to test which pH is best for our hypothetical enzyme. So here I've set up multiple test tubes, each with a different pH running from 2, which is very acidic, to 13, which is very basic. I'm going to treat everything else about these test tubes identically. So into each test tube, I'm going to put the same amount of substrate and the same amount of enzyme. Then I'm going to set a timer, we'll say 15 minutes. And at the end of that 15-minute window, I'm going to measure how much product is in each test tube. Now I assume at the beginning there was 0 product, so I'm interested in how much product happened at the end of the experiment. Imagine I collected this data. So for each pH, the amount of product varied. And this tells me that the enzyme wasn't functioning at all at low pHs, but it's kind of difficult to visualize until I graph it. If you like looking at numbers and tables, good for you. If you like looking at visualized data on graphs, good for you. You can get the same information from either strategy, but I just prefer graphs. So here on this graph, I can clearly see the range in which my enzyme appeared to be functioning, because that's the only scenario in which product was detected in my test tubes. This range is called the range of tolerance for this particular enzyme. So if we go back to the table, I've highlighted it in teal. The enzyme appeared to be functioning in the range of pH 7 through 13. Let's do another experiment where we have test tubes of different temperatures, treating them the same, waiting our 15 minutes, then detecting the amount of product in each tube. Imagine we collected this data. And again, I've visualized the data on a graph. What is this enzyme's range of tolerance to temperature? Hopefully you were able to assess this. The enzyme appeared to be functioning between the temperatures 20 and 70 degrees Celsius. So that's this enzyme's range of tolerance to temperature. It seemed to function optimally at temperature 50 degrees Celsius. That's the peak of the graph. But its entire range of tolerance is wider than that optimal point. It's this full range from 20 to 70 degrees, outside of which we notice the graph is flat and there's no product detected in those very hot and very cold test tubes. At these extreme temperatures, it's possible, well, it's likely that the enzyme denatured. Here's what you've learned. Enzymes regulate chemical reactions. Since enzymes are made of proteins, they're susceptible to being denatured in extreme environments. You also know that each enzyme has its own range of tolerance to different temperatures, pH levels, and salinity. You know how to discover the range of tolerance of an enzyme via experimentation. And in order to visualize this range of tolerance, you learned that you can graph the amount of product made under these different scenarios. Thanks for watching. If you found this video helpful, please feel encouraged to check out other videos in this series. Here is my contact information, mworkman@pima.edu. I encourage feedback, and best of luck in your biology studies.